Okay, in this session, I'm going to tell you about antibodies. I'm going to tell you how they are used in cell biology experiments, why they're so important to studying proteins. But I'm also going to show you how they are made, where we get them from, how we can create antibodies to specific proteins. But also I'm going to give you a bit of the immunology background as to how antibodies are naturally produced by B cells and how they can effectively recognize a single protein. So antibodies are the thing that we use in cell biology probably more than anything else. They're a crucial part of our little toolbox that we have to study individual proteins. So I'm going to cover how they are made naturally. I'm going to cover different types of commercially available antibody. These are the monoclonal antibodies and polyclonal antibodies. We're going to look at how monoclonals differ from polyclonals and why monoclonals are generally preferred over polyclonal antibodies, but all this will make sense later on when we see how the two different types of antibodies are made. We're going to look at the two ends of the antibody, the Fc and the Fab portion. So the Fc is not the bit that binds a specific protein. The Fab region is the bit that binds a specific protein or a specific epitope. We'll talk about species specificity when we look at applications such as immunocytochemistry, which is a technique to look for a specific protein within cells. And as we go on through the module, we're going to look at different ways that antibodies can be used experimentally in different situations, particularly when you get onto the flow cytometry lectures and when you get onto confocal microscopy, we'll be talking about using antibodies routinely. So firstly, what is an antibody? And I will refer to them as antibodies. I may also refer to them as immunoglobulins. I'm talking about the same thing. Immunoglobulins, antibodies, they're proteins. They're quite large, 150 to 170 kilodaltons. So that's 170 kilodaltons is 170,000 relative molecular mass. And a single antibody will recognize a single specific protein or chemical or epitope. It doesn't have to be a protein, it can be any moderately sized thing. So you can get antibodies that will recognize relatively small chemicals such as steroid hormones with a relative mass of a couple of hundred, or you can get antibodies that will recognize one small portion or epitope of a large protein. So naturally, antibodies are an important part of the acquired immune system. So antibodies, as you hopefully know, will bind pathogens or anything that is foreign. So anything that is not naturally produced by the host, the human body in this case, anything that enters that body that is not host could be flagged up by an antibody as being foreign. This will highlight that foreign thing for destruction by macrophages or other aspects of the immune system. Now, the cells that make antibodies are our B cells, and they mature into what we call plasma cells. And these are little antibody factories. They churn out vast amounts of antibodies. But each individual B cell will make or a B cell clone will make a different antibody to all of the other B cell clones. So as a B cell develops, it is programmed to make, it makes one type of antibody, and that it will continue to make that one type of antibody, and all of its progeny will make the same one type of antibody that will recognize one specific epitope. Now, this can occur, and we're going to go through the mechanisms of this, by alternate exon splicing of the immunoglobulin gene. So the immunoglobulin gene has got loads and loads of exons, and during B cell differentiation, some of these exons are cut out and then reshuffled and put back together to make each individual B cell able to recognize something different to all of the other B cells. So those of you who are studying the immunology and micro module, you will get this in more detail, quite possibly. But this is the basics of the system. So the immunoglobulins are these proteins, these Y-shaped proteins made up of two heavy chains and two light chains. The two light chains are shown in white, the heavy chains are shown in blue, red, yellow, and green. So these are put together from the immunoglobulin heavy chain gene and the immunoglobulin light gene. And when they've been put together in this configuration, this end bit here and this end bit here are identical, and they're called the complementarity determining regions, or the epitope binding regions, or the antigen binding regions. The most common way to refer to these end bits, which is the bit that recognizes the antigen, is to refer them as to the Fab region. And we'll talk about Fab regions later on. So this is the bit that recognizes the foreign particle. The rest of this is called a constant region because it is pretty much the same amino acid sequence amongst all immunoglobulins of that type. So all immunoglobulin Gs, which are the small soluble immunoglobulins, will have the same amino acid sequence from here all the way up to here, and on the light chain from here to here it'll be the same amino acid sequence. That's constant. It's this end bit that's hypervariable. So as your B cells are developing, and this will continue to occur throughout life, the B cells start off with the immunoglobulin gene with loads, many more exons than shown here, but you've got a whole series of exons that code your constant region, and then some V, D, and J segments. The exact numbers doesn't really matter, but there's loads and loads of them. Now, what happens as that B cell undergoes differentiation? Initially, you will get recombination of the D and the J region. So you get a cut in the DNA here and here, chop out all of these exons here, and then stick the DNA back together. And then we can get another cut somewhere in the V region and somewhere else in the D region and cut all of the DNA back together. Because we've got so many V, D, and J segments and so many opportunities to cut and stick them back together again, you can end up with almost every single B cell having a different combination of V exons, D exons, and J exons. And that makes the B cell populations, of which you've got millions and millions of B cells lurking around in your body, the vast majority of them will have a completely unique combination of V, D, and J exons that are being spliced together. That means that they will all make different antibodies. Those different antibodies are further made variable because after this process has occurred, you can then get deliberate mutations occurring to the base pairs here, causing different amino acids to be coded for in the immunoglobulin mRNAs, and then that will give even greater variation between B cell clones. Now these B cells, as they differentiate and they recombine their V, D, and J segments to create these unique antibodies, are effectively tested to find out whether those antibodies recognize self, that is, any human protein. If they recognize self, they're executed, they're killed by apoptosis, and we'll learn about apoptosis in a few weeks. If they do not recognize self, they're allowed to continue, and they are then potentially useful B cells. And you never know, that B cell that's just made it through that process may well naturally produce an antibody to COVID or something. Now, B cells don't deliberately produce an antibody to some infectious particle. Some infectious particle gets into the body, and you just hope that a B cell happens to express an antibody that recognizes that infectious particle. So to summarize, the antibody is made up of the Fab region, or the antigen binding region, also called the complementarity determining region, that recognizes the foreign particle. And then we've got the constant fragment. It's also called the fragment crystallizable, or FC region. So some people call it constant fragment FC. It actually means fragment crystallizable. Long story about why that is. I'll not bore you with it at the moment. So this is constant between all antibodies of that class. This is variable. And this is the bit that recognizes a unique epitope. So these B cells are crucial in responses to infectious agents, and they're also crucial in immunization, and they're also crucial in making antibodies experimentally. So if we want to make an antibody or collect some antibodies to a specific protein, the usual way of doing that would be to immunize an animal with that protein. So if we want to make an antibody to protein X, which is foreign to that particular animal, we will inject that animal with protein X and hope that the animal elicits an immune response against protein X. If it does, it will recognize that protein X as being non-self, therefore foreign, and any B cells and also T cells, which I've not yet mentioned, that recognize that as foreign will work together to encourage a B cell population to make antibodies to protein X. So if you've got a B cell that is programmed, just happens to be programmed to make antibodies to protein X, that B cell population will be activated and expanded, and then we'll have a large pool of B cells that make antibodies to protein X. And to cut a long story very short, we can immunize an animal with protein X, we can wait a while, we can immunize again, and then we can bleed that animal and extract those antibodies and then purify out the antibodies that recognize protein X. And that is the basic way of making a polyclonal antibody. Now, linking this together with pathogens, if you have a situation of a virus lurking around and you have a virus infected cell, you hope that there is a B cell population that just happens to make antibodies to that virus. You also hope there's a T cell population that makes a receptor that recognizes that virus. Now, the T cells, they have a T cell receptor that works in a similar way to that immunoglobulin gene that I talked about earlier, that is hypervariable. And you would hope that both a T cell and a B cell recognize the same thing. They can agree between themselves that this thing is foreign, activate each other, and then we end up with an immune response against that virus. Now this slide is more for the immunologists amongst the group, but here's a B cell, and here is its B cell receptor, which is coded by the same gene as the immunoglobulin gene. So it's basically a membrane-bound immunoglobulin, is the best way to describe it. That can bind antigen. Antigen here could be that virus or that foreign protein. If that cell recognizes that there is antigen bound here, that antigen can be presented to T cells on this MHC class 2 system, which I'm not going to go into in great detail, but that can be presented to the T cell with its T cell receptor, which itself is hypervariable. And if this antigen here is recognized by that T cell receptor as well, then this T cell can be called a T helper cell. It can enhance the survival and proliferation of this B cell population. This B cell population will be expanded because both the B cell and the T cell agree that this antigen is foreign. So this population and this population can both be expanded, and then we can get a large population of B cells which can then produce antibody, and that will be a soluble antibody based on a non-membrane bound B cell receptor is the best way to describe that. So the antibodies can float away and bind to the virus, whereas the T cell receptor can then be used to identify either virus or viral infected cells, and then those cells can be executed. So to summarize what we've covered so far, the B cells are the source of antibodies, whether we're dealing with humans or other animal systems. B cells make effectively random antibodies, which in theory could recognize anything, any chemical, any run of amino acids, any oligosaccharide, which is basically a run of carbohydrates that you often find on the outside of bacteria. Each B cell clone makes a different antibody to a different thing. However, if that B cell clone happens to make something that recognizes self, that B cell clone is executed, and all the B cell clones that don't make something that recognizes self are kept and they survive. When we get exposure to a new pathogen, virus, bacterium, something else, what we hope is that we've got enough B cells out there for one of them at least to recognize that this virus is foreign, that will recognize that it could be COVID-19 spike protein, it could be something else. We would hope we've got a B cell population that recognizes that. We will also hope we've got a T cell population with a T cell receptor that recognizes that. They will agree between themselves that this thing is foreign and to elicit immune response against it. The B cell clone will be expanded, it will proliferate, it will produce lots of soluble antibodies that will float around in blood, some of them will float around in tissues. These will stick onto that foreign target, in this case COVID-19 spike protein. That means that any infected cell will be recognized as foreign. It'll be coated with antibodies, and those cells will be recognized to be destroyed, or the T cells will directly destroy those cells. So back to the antibodies themselves, because that's what we thought we were talking about. What you've just had there is a brief summary of your immunology module, if you choose to do that module. So here's the basic structure of the antibody. They're all drawn as these Y-shaped modules, 2 heavy chains, 2 light chains linked together by disulfide bonds. This is the hypervariable region. This is the Fab region that binds the antigen. This is the constant region that doesn't bind the antigen. And the way it works is the Fab region binds the antigen, and that could be an infected cell, or it could be a virus or a bacterium. And if the Fc portion is sticking out whilst bound to a large object, that is a signal to macrophages to recognize that this Fc is jutting out from a large object, and cells called macrophages which have Fc receptors that will recognize these Fc parts of the antibody sticking out from something, and that is a signal to destroy whatever that thing is. So when we immunize an animal or a human with a foreign protein, we will get an immune response. So this is a classic chart here showing what happens during immunization and why you often have multiple immunizations. So at this time point here, we inject with antigen A, that would be protein A. And what we can see is if we assay the blood over the next few weeks, we will see a rise in antibodies that recognize protein A. We'll call that anti-A, because it's an antibody to protein A. So this takes a couple of weeks to rise, because we've got to have that antigen A or protein A in the blood or in the tissues. That will activate a B cell population that just happens to make antibodies to protein A. They will proliferate, they will secrete lots of antibody, and we will detect it in blood, and that will peak a few weeks later. Then the levels of anti-protein A will decrease until the organism gets challenged with protein A again. So a few weeks later, you can give a second dose of immunization, in this case, again, antigen or protein A again. And in this case, we get a much more potent response because A, there are many more B and T cells that recognize protein A, and that will give a much more potent antibody response. And you're much more likely to get memory cells which have a long-term memory of that antigen, so that those B cells will hang around long term because they've seen the same thing multiple times. It means that they're an important clone of cells that need to be kept long term. So you'll see that serum antibody titer is much larger at the second exposure than the first exposure. Whereas if you injected with protein B at this time point here, you get the same size response here as here, because it's the first time this animal has seen protein B. So as I mentioned earlier, if we want some antibody to protein A, the best way to get that is to take an animal, inject it with protein A, and extract its antibodies or extract its B cells. So if we want to have a rather crude, dirty antibody prep, we can just take an animal, typically take a relatively large animal, so a goat, rabbit, donkey, horse, and inject that animal with protein A. The animal will have an immune response to protein A. You can repeat the process so you get a high antibody titer, and then bleed that animal. And you tend to not kill the animal at this point, but bleed the animal and then repeat the bleeding at later time points. You then take that blood, remove all of the white cells and red cells from there, and then extract all of the antibodies from that. And then we can purify the antibodies to protein A by having some protein A immobilized onto a column and then run the antibodies down that column, and wherever those antibodies come into contact with protein A on the column, they will stick, and all of the antibodies that don't recognize protein A will just wash through the column. We then wash all the excess through with saline and then change the salt concentration to elute off the antibodies that are bound to protein A, and then we end up with a pure preparation of anti-protein A antibodies. Now, if that's done in a goat, that would be called goat anti-protein A. Now, we call these polyclonal because this will recognize every antibody that's in that animal's blood that recognizes protein A. And you think about proteins as being large globular proteins, or it doesn't matter what shape they are, they have multiple epitopes on them. There could be 20 or 30 different epitopes on the outside of a typical protein. So if you inject protein A into a goat, you might get a mixture of 30 or 40 different antibodies, all recognizing different bits of protein A. So we call it a polyclonal antibody because all the antibodies come from different B cell clones. Now there's a different way of producing antibodies, and that is to take a smaller animal. In this case, it's usually a mouse. It can be a rat, but it's typically a mouse. You take that animal, inject it with protein A, kill the animal, extract the spleen cells, which is where you've got immature B cells present. Now, once you've got those spleen cells, this is at a set time point after inoculation with protein A. You fuse the spleen cells with some multiple myeloma cells. Now, multiple myeloma is a B cell type cancer. So these are cancerous cells. So they're immortal, they can grow long term. The spleen cells would die in cell culture very quickly. So they have a very limited lifespan. So if you can fuse the spleen cells with a B cell tumour cell to create these hybrid cells, then the genetic information from the spleen cells that can make our antibodies can be propagated long term. And we call these hybridomas because they're a hybrid between a spleen cell, immature B cell, and a myeloma B cell cancerous cell. So we create all of these hybridoma cells. We isolate single cell clones. That is basically put one cell into the cell culture well, and you'll see cell culture wells next week, or you may have seen them previously. Typically 96 well plates where you put a single cell in a single well, so we call that cloning. And then we grow them up and we take the supernatants, the liquid medium that those cells are being grown in, and we assay that medium for the presence of antibodies that recognize protein A, and we can do that by ELISA type technology. So the end result of this is we've injected antigen into a mouse, we've extracted its spleen cells, fusing with myeloma cells, created hybridomas, We then plated thousands of individual cells into thousands of individual wells, allowed them to grow long term, and then we've got thousands of wells of growth medium to check to see if those wells contain anti-protein antibodies. If they do, we can propagate those cells that make that antibody and grow them permanently long term, and then produce that antibody en masse, and we can scale that up to produce milligrams of protein and effectively unlimited supplies of that protein, although it is a very expensive process. So one of the main things that we do with antibodies is use those antibodies to detect the presence of proteins or epitopes in biological samples. And one of the main techniques that you are depending on which course you're on, you may well do, which is immunocytochemistry. Another one that you may well do is ELISA. And both immunocytochemistry and ELISA can work in a similar way. Now, for immunocytochemistry, this is using antibodies to detect antigen in cells. If it's immunohistochemistry, it's detecting antigens in tissues. So imagine that we have got an antigen, and let's say this is the surface of a cell stuck on a microscope slide, and this is the antigen on the outside of a cell. We would have a primary antibody which recognizes the antigen. So this could be protein A, and this is our primary antibody that could be goat anti-protein A. If this was a polyclonal antibody, if it was a monoclonal antibody, this would be mouse anti-protein A. So we have our sample prepared, we apply the primary antibody that recognizes the antigen, allow that to bind, wash off the excess. We then apply a secondary antibody, and the secondary antibody recognizes the first antibody. So if the first antibody is goat anti-protein A, we need an antibody that recognizes goat immunoglobulins, specifically the Fc portion. So what we could do is take the Fc portion of a goat immunoglobulin, inject that into a rabbit, extract the antibodies that recognize goat Fc from the rabbit, and then apply that antibody at this point, and it will bind wherever the primary antibody has bound. Now, the secondary antibodies tend to be conjugated with a detection system. And in this case, it's horseradish peroxidase. And the horseradish peroxidase is an enzyme that can be tagged onto antibodies, and that will turn a substrate, in this case, a substrate called DAB, and it will turn it from a colourless to a dark brown or black colour. So what will happen is you'll get a colour change reaction wherever protein A is on your sample. The primary antibody will bind. Wherever the primary antibody is bound, the secondary antibody, the rabbit anti-goat Fc, that will bind. That brings in the horseradish peroxidase enzyme, and that creates a colour change reaction. So the secondary antibody is explained here. This is made by immunizing an animal with the Fc portion of an antibody from a different species, the species with use for our primary antibody. So in all of these systems, you have two different animal systems. You have one animal that produces the primary antibody. You have a second animal that produces the anti-Fc of the first animal, and you use both antibodies in these systems. Now, when I say you could inject an animal, well, in reality, somebody has injected the animal, but we just go to a catalogue and we can buy antibodies to almost every single known human, mouse, rat, rabbit protein that there is known. You can just buy them from a catalogue. So in my experiments, I will buy an antibody to one of my favourite proteins, let's say P53. I can buy a rabbit anti-P53 protein off the shelf. It'll cost me about £200 for 100 micrograms. I can then buy a goat anti-rabbits Fc antibody for another £150, and that will be horseradish peroxidase conjugated for me. So I can just buy all of these things off the shelf and do my experiments. But since I've talked about both polyclonal and monoclonal antibodies, there are reasons why we use them in different situations. So here's a polyclonal, here's a monoclonal, and these are various properties. So the supply is limited and variable. If you make a batch of anti-protein A antibody from a goat, well, you can only bleed that goat a finite number of times. If you need more of that antibody, you've got to inject a different goat. When you do that, you might get a different immune response with a different animal, so you get a slightly different batch of antibody. So if you want to do experiments over a long period of time, polyclonal antibodies, they can differ from batch to batch. But they are low cost and they are relatively low specificity compared to monoclonals, which I'll come on to shortly. Now, monoclonal antibodies, although these are produced by injecting mice with, let's say with protein A, technically unlimited, because you can fuse those B cells with myeloma cells to produce the hybridomas and grow them en masse. So if you want to create a new batch of anti-protein A antibody, well, it's going to come from the same B cell population. It's going to be exactly the same monoclonal antibody every time you make it. So there is no very limited batch to batch variation. It's the same immunoglobulin class or subclass. Now we have IgGs, immunoglobulin G, which is your main soluble immunoglobulin. There's IgG1, IgG2, IgG kappas, all sorts of different subclasses. They're always exactly the same from a monoclonal antibody prep, whereas a polyclonal antibody, you get a whole spectrum. The costs are actually quite high. You're talking about £1 million a gram. And specificity is very high. And it's very high because each antibody is programmed to recognize one epitope, whereas here you might have got 40 or 50 antibodies all binding to the same protein. If you have a single antibody species, then you can assay to find out if that antibody recognizes something else, and if it does, you throw it away. You work on antibodies that only recognize a single epitope. Whereas with these polyclonal antibodies, you could have 30, 40, 50 different immunoglobulin proteins in there, all recognizing the same thing. And if you've got 30 or 40 of them, there's an increased chance that one of those might also, by chance, recognize something else. So so polyclonals, by definition, have a lower specificity than monoclonal antibodies, and therefore monoclonal antibodies are very much preferred wherever you can get hold of them. So in a typical experiment where we're detecting proteins within cells, you can either have a cell suspension or it could be a cell attached to a microscope slide. And this would be thousands of cells, not just one cell. We then fix that cell, typically in paraformaldehyde. We can permeabilize it with alcohol to punch holes in the membrane. That allows the antibodies to get inside the cell and bind to the antigens if it's an intracellular epitope that we're looking for. If it's an extracellular epitope, this is not such a problem. What we then tend to do is do a counter stain with something like hematoxylin. Hematoxylin is a nuclear binding stain, or DAPI, which is a nuclear fluorescence stain. And we look for the colours because we can tag the antibodies with either a chromogen. I've mentioned horseradish peroxidase turning DAB into a black colour. Well, we can also tag these antibodies with fluorescent molecules, red and green, and almost any colour that you wish. And this slide shows a typical immunofluorescence immunocytochemistry. So that's using fluorescently labelled anti-F actin. So in red is where the F or filamentous actin is present. Green is where tubulin is present. And then blue is a chemical called DAPI, which just stains the nucleus fluorescently blue. And using a fluorescence microscope, you can get a 3-colour image to see the presence of these 2 different proteins in relation to the nucleus. So this is what we call immunocytochemistry. Now in future lectures you'll come across flow cytometry, where we perform this process but have cells in suspension, and instead of passing them through in front of a microscope lens, we pass them in front of a laser and interrogate them that way. You'll learn about that in a few weeks. And then confocal microscopy, you'll learn about that in a few weeks as well, which is usually performing immunocytochemistry but using a special type of fluorescence microscope that can illuminate just a single layer of the cell, and we call that Z stacking, but I'll leave that until Sue Campbell's lecture in a few weeks' time. Okay, finally, I'll just add one final point as ELISA technology. ELISAs are where we use antibodies to detect typically soluble proteins or epitopes, and we can have antibodies bound to a plastic multi-well plate. We can then add antigen to that, and then we can detect the presence of that antigen with a second antibody. There are various different ways of doing ELISAs. You'll come across them at some point during your PSP2 labs. So I'm not going into them in detail now. They might also be covered in MBB. Okay, so that's the end of this video.